Well, good afternoon, everyone. It is 12 o'clock, so we are going to get started. I am thrilled to introduce Dr. Krishna Komanduri. He is an international leader in the fields of hematology oncology, transplantation, and cellular immunology, and the Division Chief of Hematology Oncology at UCSF Health. Dr. Komanduri also acts as the physician-in-chief of the Helen Diller Family Comprehensive Cancer Center, as well as the clinical director of the UCSF Living Therapeutics Initiative. As you'll learn, Dr. Komanduri is also a product of UCSF, having completed his fellowship here and several years as a junior faculty member before he was recruited away to be a physician scientist at MD Anderson. Since that time, he's become a national leader in the field of hematologic malignancies and transplantation. He's served as president of the American Society for Transplantation and Cellular Therapy, the major professional society for his field, and he's chaired the American Society of Hematology Scientific Committee on Immunology and Host Defense, and he serves on the board of directors of the National Marrow Donor Program. Krishna's own research, which is both lab-based and translational, focuses on the improving outcomes of patients undergoing transplantation for hematologic malignancies, as well as the use of immune-based therapies for all cancers. He's authored or co-authored 6 books and 125 articles in the peer-reviewed literature. And he's been a PI or key investigator on more than a dozen grants and has served as a member of several NIH study sections. We are absolutely thrilled to have him back at UCSF and welcome him in giving our grand rounds today. I'll turn it over to Krishna.
Thank you so much, Michelle. It is a genuine pleasure to be back at UCSF and especially to present at this wonderful educational conference. And I want to thank you and Dr. Walker and Lakshmi for inviting me. I'm going to share my screen here. I hope you can see that. Here are my disclosures. So it has been 23 years since I left UCSF, and I think it was an interesting milestone to me that I started on July 1st as the division chief, exactly 25 years from the day that I finished my fellowship. So a lot has happened in 25 years. In 1997, I transitioned from UCSF fellow to a junior faculty member. Mark Zuckerberg had a Star Wars themed bar mitzvah. Princess Diana was killed in a Paris car crash. Some of us remember watching that event. I lived in a 1-bedroom apartment with a view of the Palace of Fine Arts in the marina that I paid for, which I rented at $900 a month. And critical to what we're going to be talking about today, allotransplant was usually done only in patients under the age of 60 and had a 30 to 40% 100-day mortality. And there were effectively no good options for patients without a well-matched donor. So obviously, I'm thrilled to be back here, and 25 years later as the division chief. I'm still having a bit of imposter syndrome. Of course, now San Francisco General Hospital is now named ZSFG, and I'm still not used to saying that. Diana's ex-husband has ascended to the throne, and I'm not even going to get to the issue of what San Francisco real estate has done in that time because it depresses me. But I should say, allotransplant now, I'll talk about how much our outcomes have improved with respect to both early mortality and donor options. So allogeneic transplant, and I'm really not going to focus broadly on all kinds of indications like, for example, bone marrow failure diseases or sickle cell disease, all diseases that can be cured with allogeneic transplant. I'm going to focus on leukemias. This is a painting of Alfred Valpeau, who was a French physician. And this is supposed to depict the incident where he diagnosed leukemia in a French florist. This florist came in with a very pale complexion, a very massive spleen, and Valpeau described what he called a gruel-like consistency of the blood. And this was again in 1825. And later in that century, others like Rudolf Virchow and at the end of the 19th century, people like Paul Ehrlich, who developed supravital dyes to look at blood and acid-fast bacilli and others, really helped us to define the pathologic diagnosis of acute leukemia. However, there was really no clinical progress. In the first 125 years in the leukemia therapy, there were absolutely no cures. And indeed, in those 125 years, there were really only 1,000 publications about leukemia, and they were really all diagnostic. From 1950, when people started to treat this with alkylating agents that were a product really of wartime and the known effects of mustard gases on bone marrow, there were 175,000 publications about leukemia. So you can see a massive explosion. But it was really in the 1960s that we started to use combination chemotherapy and stem cell transplants. The first breakthroughs came with intensification of chemotherapy. And those of you who've read the book, The Emperor of All Maladies, will know that that first chapter is riveting. Jay Freireich and Tom Fry and others at the National Cancer Institute really started to first have the result of prolongation of life in children with pediatric leukemia who at the beginning of the 60s lived typically a few weeks to a few months. And they started to see long survivals in these patients, however, few cures from 1968 to 1970. Since then, this is actually one of the most remarkable, I think, collective efforts in iterative improvements in modern medicine. And this is just to 2009, but we now regularly expect survival rates in pediatric ALL patients at 95% or so. But the general principle that Freireich and Fry advocated was really more intense and longer chemotherapy. So when we started to do stem cell transplants, and Don Thomas started to do stem cell transplants, for which he shared a Nobel Prize later on with Joseph Murray in 1957, but it wasn't until 1968 that the first curative transplant took place. The sad thing is that we really knew nothing about immunology at that point. And in the 1950s, Arnold Rich remarked that literally nothing of importance is known regarding the potentialities of small lymphocytes. And he called this one of the most humiliating and disgraceful gaps in all medical knowledge. Again, in that decade, in the 1960s, Jacques Miller first defined the role of the thymus as a producer of T cells, something that was not known until then. And in 1968, along with Robert Good, who was at the University of Minnesota, proposed the existence of separate lineages of B and T cells. And since this is a family grand rounds, I won't read this, but this is what was said at a major national meeting, that B and T are the first and last letters of, let's say, people were skeptical. It wasn't until the 1970s that we characterized separate subsets of CD4 and CD8 T cells and really understood the mechanisms of antigen recognition. And indeed, one of the prouder moments of my fellowship was taking part in a team science project that was led by Mike McCune and Rafik Sekali in Montreal. We demonstrated for the first time that human thymic function actually persists in adulthood, even late into life, which was something that I was taught in medical school did not happen, that the thymus was a vestigial organ. So HCT, hematopoietic transplantation, expanded without a good understanding of immunobiology, which was really essential for its success. But this became critical when graft versus host disease was just recognized as a major complication. When we realized that T cells from the donor could attack the recipient, causing graft versus host disease, in the 1980s, people started to do trials of T cell depletion. And you can see here relapse rates in individuals who had a standard transplant here, and for example, didn't have graft versus host disease, or got a T cell depleted transplant. Basically, the relapse rates doubled. These were studies that were done in identical twins who, of course, don't have any alloreactivity and can't get graft versus host disease. And they also had roughly 50-ish% relapse rates as opposed to 25%. So this told us that the T cells that were in the donor graft were not just passive and irrelevant to the outcomes. We thought that the chemotherapy and radiation that we were giving was really the essential part of the transplant, but indeed that the T cells were mediating relapse. And when you took them out, relapse rates effectively doubled. So that led us to really, our common, I would say our modern understanding of what is happening in an allogeneic transplant. We can get graft versus host disease, which is mediated by T cells, typically naive and early memory T cells. We have tumor reactive T cells that cause what we call graft versus tumor responses or graft versus leukemia. And we also have T cells transferred from the donor that protect the recipient from pathogen-specific immunity. So if we move along this timeline, we really now started to see curative therapies, including chemotherapy and stem cell transplants at the end of the 1960s. But it wasn't until the 1990s that we realized that T cells were critical for transplant cures, and there was a dramatic increase in success. And this just shows you, in a 10-year period, how much stem cell transplant changed. And these are reduced intensity, the consequences of using reduced intensity or lower chemotherapy doses to really facilitate the immunologic effects of the transplant. So if you look at unrelated donor transplants facilitated by the National Merit Donor Program, there were almost none in 2004 done in patients over the age of 64. And remember, this is important because most leukemia patients actually are over the age of 64. And you see a massive increase to between 2004 and 2014. And you see this in all age groups except really a much more modest change in younger patients. And you can see that we're now addressing the need in patients who actually have the highest incidence of acute myeloid leukemia. So where are we? We have evolved since the 1980s from an ablative approach where we think that we're giving super high doses of chemotherapy and curing patients that way, to recognizing that we're really doing immunotherapy. And therefore, the use of this less intensive conditioning expanded the eligibility. We couldn't do transplants in people over the age of 55 or 60 because of comorbidities. Now we routinely do transplants, allogeneic transplants, to the age of 75. The use of other modalities like peripheral blood stem cell transplant and improved supportive care and nursing care have substantially decreased relapse-associated mortality, also infectious disease care, I would say, from 30 to 40%, as it was when I left UCSF, to now 5 to 10% in the first 100 days of allotransplant. So typical results for AML, we have 5 to 10% 100-day mortality and 30% one-year mortality. So there is still mortality, and unfortunately that mortality is about 50% due to relapse and 50% due to non-relapse mortality, things like graft versus host disease and infection. So GVHD is still a major problem. In acute forms, which happen 10 to 30% of the time in a severe fashion, and chronic forms, which happen much more commonly, 20 to 70% of the time, and are a major barrier to quality of life. Sadly, in allogeneic transplant and in other fields, immunosuppression is only modestly improved and is still largely non-selective. There are only 3 GVHD therapies approved in the last 50 years: JAK inhibitors, ITK, BTK inhibitors, and ROCK2 inhibitors. And this is a major limitation in the field. So a key question is, how do we selectively inhibit these cells without impairing graft versus tumor responses or pathogen-specific immunity? So these are some of the approaches. Again, we have only those 3 approved therapies. These are some of the things I'm going to talk to you about today from a biological standpoint: post-transplant cyclophosphamide, inhibition of T cell signaling, and ways to in vivo expand regulatory T cells. So again, why do we care? Well, if we don't have a well-matched sibling, and again, each sibling has a 1 in 4 chance of being a perfect match, we have to go to unrelated donors. But if you don't have a perfectly matched unrelated donor, then you end up with outcomes that successively fall off. So with one antigen mismatch, you have about 10% worse survival. And this is a classic paper by Stephanie Lee at Fred Hutch. And with a 2 antigen mismatch, you have really almost about half the survival that you would have. And so historically, we have not done unrelated donors with more than 1 mismatch because of this fall off in outcomes. The problem is this leads to disparities. So if you are Black or African American, you have only a 29% chance of having a matched unrelated donor in a registry. If you're Asian or Pacific Islander, about 50%. But if you're white, Northern European, it's about 80%. Well, what if can we just register our way out of this problem? Can we just go to every place and knock on every door of every Black and African American donor and just make sure that we have enough of them? Well, we have a 29% match rate with current registry members, but if we took all eligible donors, that is, of suitable age and comorbidities, at the current acceptance rate into the registry, because not everybody will agree, we would have a donor for 45% of all Black patients. But if every donor said yes, which of course is unreasonable, we would still only be able to match 64% of patients. So a huge disparity gap remains. So we need alternative donor types. The US is becoming more racially and ethnically diverse. And this a diversity index that measures probability that 2 people chosen at random will be from 2 different racial and ethnic groups. And indeed, look where we have the greatest diversity index in 2010, it's in California. And this has increased from 55% to 61%. So we're getting more diverse. And interestingly, this is not something I realized until, again, serving with the National Merit Donor Program, because we have more mixed race individuals like my own children, it is getting more difficult to match over time. So patients 60 and over will have a registry match 54% of the time. But our youngest individuals, like my children who are under 20, have only a 34% chance of matching broadly, not even as individuals. But mismatched graphs, the ability to use mismatched graphs could close this disparity gap. And again, this is informatic modeling by Be the Match, or the National Merit Donor Program, which runs the registry. If we could do successful 7 out of 8 transplants or 1 antigen mismatch, 72% of African-American patients would have a donor. If we could do 6 out of 8 transplants, 97% would. And if we can do 5 out of 8 transplant, effectively everybody in this group, which is the least likely to be matched group, would have a donor. So how are we doing this? Well, one way, of course, is to inhibit alloreactivity after the transplant. So Leo Lesnick and Ephraim Fuchs in murine models and then in human translation started using post-transplant cyclophosphamide to facilitate haploidentical or half-matched transplants. So the simple part of the slide is that there are donor T cells that don't recognize the recipient. And they, after being infused into the recipient, are quiescent and not rapidly dividing, as are memory cells that are seeing pathogens that aren't actively being used and activated early on. But the cells that recognize the recipient get rapidly activated immediately after they go into the patient. So if you give chemotherapy, traditional chemotherapy, which is cheap, cyclophosphamide, on day 3 and 4, it deletes these activated cells preferentially. Now, of course, some of these get affected too, but largely it is selective. So the National Merit Donor Program, working with transplant centers, did a multi-center study, and I'm proud to say that at the University of Miami, of the 80 patients treated on this study, that we were the #1 accruing center. We enrolled 21% of the 80 patients, and Antonio Jimenez, who's a junior faculty member who I recruited to Miami, was the co-first author of this paper published in JCO last year. The primary endpoint of the study, which was exploratory, was to see if 1-year survival using this approach, using mismatched unrelated donors and giving post-transplant cyclophosphamide, could lead to outcomes similar to what we expected. And I told you that we expect typically 1-year survival at about 70%. And while this is not a randomized study, we saw 72% and 79% 1-year survival, depending on the type of conditioning given. So this is highly, highly encouraging. Well, at the University of Miami, we became, because we have a highly diverse population, just like we do in San Francisco, the leading mismatch unrelated donor transplant center in the US, based on the patients that we did on protocol, as well as patients we did off protocol who didn't qualify. So we performed a retrospective study of 73 of these patients who all had mismatch unrelated donor transplant. Roughly half received this new approach of post-transplant cyclophosphamide. The other half, or 34 patients, had this historical approach of using antithymocyte globulin, which was given with the conditioning. And importantly, 70% of our population that we looked at were Hispanic and Afro-Caribbean patients, so patients who really don't often have registry donors. And 30% of them on the experimental arm, which is the PT-Cy arm, had highly mismatched grafts, that is more than 1 antigen mismatch, so we wouldn't normally have done a transplant. So Antonio Jimenez reported these retrospective results, and you can see in the red, the PT-Cy arm, and you can see that there is much higher overall survival and much lower non-relapse mortality compared to the historical approach of using antithymocyte globulin, which we did. And note that these are not actually equally risky groups. These groups had only 1 antigen mismatch, and these groups had, again, 30% who were highly mismatched, that is 2 or 3 antigen mismatches. So despite being at much higher risk for graft versus host disease, they did much, much better. So prompted by these results, Antonio and I worked with 2 close colleagues, Miguel Perales and Brian Schaefer at Memorial Sloan Kettering, and these results were just published last month in Blood Advances. We looked back again at post-transplant cyclophosphamide and ATG at our centers, and again, GVHD-free, relapse-free survival, overall survival, relapse, and non-relapse mortality were all significantly better. So this really, again, highlights, even though it's a retrospective study across almost 150 patients, that this really appears to be an exciting and very viable approach. We're critically excited that the NMDP has decided to do a much larger study, not only using adult patients getting bone marrow, but also using peripheral blood stem cell grafts. but also having older patients getting a reduced intensity graft, and for the first time doing an exploratory group of pediatric subjects, which we didn't want to do the first time around. The primary endpoint is 1-year overall survival in each of these 2 adult cohorts with an exploratory analysis in pediatric patients, but there will be 180 patients treated. The good thing is the study is so exciting that it's well ahead of accrual targets. We were hoping to be the lead accruing center at the University of Miami. We're the 3rd accruing center, but there are 16 patients treated there so far with 10 patients in the queue. City of Hope and Memorial Sloan Kettering are the 1st and 2nd leading centers at this point. But this study will close early, which is very unusual. And I should say 50%, over 50% of the patients treated are minority patients, and that's very unusual. We're lucky typically if we get 10% of patients who are in underrepresented groups on clinical trials like this. So I'm going to talk about some biological approaches that we've worked on in my lab. As Michelle noted, that I've really been a translational immunologist, but I consider myself a laboratory-based scientist at my heart. So again, the critical question: how do we not inhibit graft versus tumor responses or pathogen-specific responses while inhibiting graft versus host disease? So this is a bit of a flashback, not a slide that I normally show, but I'm very proud of this because it's work that I did as a Hem-Onc fellow and a first-year faculty member in Mike McCune's lab at the Gladstone Institutes of Virology and Immunology. Mike remains a friend and a mentor and an inspiration to me, and had a long relationship here in the Department of Medicine. So we developed at that time an approach that was then novel, to look at antigen-specific T-cell responses and measure them by stimulating peripheral blood mononuclear cells from patients, and then looking at intracellular cytokines by flow cytometry. So here we're gating on CD4 T cells and then looking inside those cells, looking at an activation marker positive and intracellular cytokine positive following stimulation with CMV antigens. And I won't go through the details of this report, but working with Mark Jacobson, who was an infectious disease faculty member here, and others, we showed, I think one of the first demonstrations that antiretroviral therapy in the HIV setting could lead to immune reconstitution of CMV-specific T-cell responses, even in patients who had active CMV retinitis and encephalopathy. So this technique we validated as being clinically relevant, and indeed, cytokine flow cytometry is now widely and primarily used to assess the frequency of virus-specific T-cell responses, cancer vaccines, et cetera. Later, adapting these approaches to get more and more sophisticated, looking at multiple cytokines, we looked more at CMV-specific T-cell responses, and Tae-Kon Kim, who was a PhD student in my laboratory, now a faculty member at Vanderbilt, and I showed that most CMV-specific T-cells are actually now what we understand as being late memory cells. So when cells come out of the thymus, recognize an antigen, they go through differentiation stages and become progressively differentiated. And indeed, of that maturation spectrum, CMV-specific cells are highly differentiated, not naive or early memory cells, but highly differentiated. Well, why does this matter? Well, we can look at signaling in T cells, and indeed, signaling pathways, including the calcineurin pathway, are exploited by using calcineurin inhibitors like cyclosporin A or tacrolimus. The mTOR pathway is active in T cells, and we can inhibit that using sirolimus or rapamycin. But a pathway that really hadn't been explored in T cells as a way of doing immunosuppression was the MAP kinase pathway. We think of MAP kinase pathways being important in malignancies and in other settings, and indeed, there are a number of MEK inhibitors, including selumetinib and trametinib, cobimetinib, that have been improved in the setting of melanoma and are being used in other disease settings as well. So we decided to see whether we could use MEK inhibitors as a way of inhibiting T cells to try to achieve more selective immunosuppression. So this is work, again, that was published by Tae-Kon Kim, who was a graduate student in the lab, and Takeru Shindo, who was a postdoc in the lab. Takeru is a faculty member now in Kyoto, and again, Tae-Kon is at Vanderbilt. Eric Weider was actually with me at UCSF at the Gladstone, and along with Kara Benjamin and Robert Levy. So Tae-Kon initially showed, as part of his PhD thesis, that as we looked at naive and late memory cells, if we look at the amount of ERK activation that is downstream from the RAS-RAF pathway, we see more utilization of this pathway that is higher mean fluorescence intensity in naive and early memory cells. Well, this was exciting to us because we knew from the work of other people, including Warren Schlomchik then at Yale and Steve Emerson, that naive and early memory cells were actually the most important cells for graft versus host disease. And these late memory cells, as we showed before, were actually really critical for pathogen-specific responses to viruses important in the transplant setting, like CMV and EBV. So we thought, well, maybe if we inhibit the MAP kinase pathway, we'll preferentially inhibit alloreactivity and relatively spare virus-specific T cells. Since these data are published, I'm not going to go through the details, but this is an editorial that was published by Paul Martin of the Fred Hutch that accompanied our article. And basically what we showed was that ERK phosphorylation decreases with increasing memory differentiation, that alloreactivity was here and pathogen protection was here. So by giving MEK inhibitors, and we showed this both with human cells and then in murine models, we could preferentially inhibit alloreactivity and in vitro, for example, inhibit alloreactivity highly effectively, better than we could with tacrolimus in vitro, while leaving CMV-specific T cells alone, which didn't happen when we used those traditional immunosuppressive agents. So to get to some unpublished work related to this, so based on this paper in Blood by Takero and Tekon, we suggested that inhibition of the MAP kinase pathway could prevent graft versus host disease. And I already showed you that administration of cyclophosphamide, this PT-Cy approach, can prevent but not treat established graft versus host disease. Again, Ephraim Fuchs and Leo Lesnick at Hopkins had done trials to try to treat early graft versus host disease, and it didn't work. So we hypothesized that maybe using PT-Cy at this late stage, along with MEK inhibition, could treat early onset graft versus host disease without impairing relapse. And our rationale was the following. The other day, I heard about a fire in an Oakland hillside, and I heard on watching local news that the firefighters were there all night because they were worried about the brush fire flaring up after they had put it out. Well, why? Because you have inflammation, you have tissue injury, and even if you knock out the T cells that cause that, you can then prime new T cells that basically restart the fire. So we thought that while PT-Cy alone didn't work, chasing with a MEK inhibitor, which is again could be administered orally and very safely, could prevent that reflaring of graft versus host disease. So indeed, George Nahas, who actually had an ASCO-YA award with me, was a fellow, he's now a faculty member elsewhere, showed that basically if we administered post-transplant cyclophosphamide at day 12 and 13, followed by trametinib for 8 to 14 days, we can get 100% survival of all of the mice, just like a bone marrow transplant that was done without any T cells. And again, significantly better survival. We had, again, more than 50% death of the mice in this model if we gave either cyclophosphamide alone or, again, at this stage, tried to give a MEK inhibitor. George also did animal studies to look at the graft versus tumor responses. So if you have bone marrow only and no cancer, of course, you have no tumors appearing in the mice. If you have bone marrow and then give cancer cells without any T cells, the mice all die because of the cancer. If you give bone marrow and T cells and the lymphoma cells, the mice live here, but if you follow them out longer, they die of graft versus host disease. And what we found, again, here, and these are data that are being prepared for publication, is that if we gave bone marrow and T cells that mediate the graft versus leukemia responses and post-transplant cyclophosphamide and trametinib, we now keep the mice alive and they don't get killed by leukemia. So we have intact, we have successful graft versus lymphoma responses. So to summarize these studies, MEK inhibition may selectively target alloreactivity while sparing pathogen-specific immunity. We have evidence of a class effect. We've done this with multiple MEK inhibitors. Immune recovery may be protected in humans by more selective inhibition. Hidekazu Itamura, who came to my lab as a postdoc, showed in a JCI Insight paper that MEK inhibition didn't impair donor regulatory T cell function. Again, we have again shown in unpublished data that PTCy followed by MEK inhibition is effective in established graft versus host disease. So another exciting approach is expanding regulatory T cells. Well, we know that here, Jeff Bluestone and Shizhi Tong and Mark Anderson and others have shown that we can expand regulatory T cells and use them to perhaps to fend off diabetes or treat autoimmune disease. So we worked on another approach to actually expand in vivo regulatory T cells. So there's a pathway called the TNF receptor superfamily 25 pathway. Its only ligand is a protein called TL1A. This pathway can be activated via an agonist antibody or via a fusion protein. And both of these reagents were developed by the late Eckhard Podack, who was the chair of microbiology and immunology at the University of Miami. And the expansion of the target cells could expand effectors or regulatory T cells depending on their activation state and is IL-2 dependent. So Robert Levy and I and Dietlinde Wolf, who worked in my lab and bridged our two labs, did studies in mice where we gave this TL1A fusion protein to mice that were healthy. And regulatory T cells are normally 10% of the peripheral compartment, but we found that giving this TL1A Ig fusion protein and low-dose IL-2, we can expand regulatory T cells to 80% of the peripheral compartment in the mice. The mice were unaffected. We followed them for 6 months and they were had no other consequences. But then, and that comes back down naturally. So if we, after the expansion, now do a transplant into recipients, we wanted to see if we did that transplant across the mismatch barrier, what happened to the mice. And the bottom line is that it works beautifully. Mice given TL1A Ig, that is, we do regulatory T cell expansion from the donors, and now transfer cells that would normally kill the recipient. All of the recipient mice survive. Graft versus tumor responses are intact, and they look exactly like mice that didn't receive any T cells at all. So this is pretty exciting, but it would mean potentially, if we translate it, expanding using this kind of reagent, regulatory T cells in a donor. And obviously there are more safety concerns in that setting. So we're proceeding cautiously. Murine studies in monkey have been done. Monkey studies are actually planned, and we hope to translate this to human studies. In comparison to post-transplant cyclophosphamide, when we do a regulatory T cell expansion in the donor, the thymus recovers much more robustly in recipients, and other measures of immune reconstitution are better. This is published by Dietlinde and Robert and I in JCI Insight a few years ago. Another exciting approach is mesenchymal stromal cells. These were first shown in 1995 to have potential clinical potential in steroid refractory graft versus host disease. There are a lot of potential applications. And unfortunately, a mesoblast study in pediatric GVHD was the only study that was positive, but in other settings, infusion of intact mesenchymal stem cells wasn't effective. Mesenchymal stem cells, if you give the entire cell, are context dependent, and after infusion, traffic to the lungs and then redistribute. And effective dosing and redistribution may be difficult because of their size and other problems. But we think the activities of MSCs are actually mediated by secreted proteins and other, again, circulating molecules. So Van Badiavis and I, who is a dermatologist at Miami, who is interested in mesenchymal stem cells and vesicles derived from them for wound healing, decided to do experiments looking at these extracellular vesicles in murine models of transplantation. So these extracellular vesicles and the proteins secreted from them were really thought to be trash. They're found in essentially all biological fluids, and they contain molecules from the host cell, including RNA, DNA, lipids, and proteins. They can exert effects locally and distantly. They're far more stable than cells, and they can be produced at scale very, very easily. So we did animal models where we gave bone marrow either no T cells without exosomes, or T cells plus exosomes. And you can see here, if you give exosomes at high dose daily and do a transplant that would normally be very rigorous, would kill all of the recipients due to graft versus host disease, all of the animals survive, where if we give T cells without exosomes, all of the animals die. If you look at the skin pathology, this is a control animal. This is a graft versus host disease animal. You see a lot of sclerosis and thickening. And you can see that a lot of that is improved in the recipient animals that have received extracellular vesicles. Same thing with the gut on the right. You can see that the tissue architecture and the villus destruction that happens in graft versus host disease is dramatically improved when we give EVs in these murine models. And this also is being commercialized. We hope Van has started a spin-off company called Agley that has 2 INDs for wound healing, and we hope to move this toward clinical therapies in graft versus host disease. The last thing I'll say about graft versus host disease is that location matters. Trent Wang and Anat Galor at the Bascom Palmer Eye Institute. Trent runs a graft versus host disease and a junior faculty member in my old program, is now looking to see if we can pull T cells straight off of the eye, basically by a Scotch tape-like approach, basically where we do imprint, and then wash those cells and look. And these are just one healthy donor in an ocular graft versus host disease patient that he recently looked at. And you can see that the ocular graft versus host disease patient, and we wouldn't normally be able to otherwise diagnose this in other ways, has lots of T cells on the cell surface. So we're really interested in looking at this, seeing if this could be an early marker, because often by the time people develop significant eye graft versus host disease, it's not reversible. So perhaps migration of T cells to the eye could be an early predictive or prognostic biomarker. So the last part of my talk, I'm going to shift gears completely and talk about CAR T therapies, because this is really, I think, the most exciting area of cellular therapy. We will continue to do allogeneic transplants, especially for relapsed leukemias, and especially myeloid leukemias, where we don't have CAR T cell therapies under development much. But CAR T therapies have revolutionized lymphoma therapy. This is a lovely video that Stan Riddell at the Fred Hutch gave me, and I'm going to see if this works. So these little rounded cells are CAR T cells, and these are glioblastoma cells in a dish. And I just love to look at this video because the glioblastoma cells are being destroyed by the CAR T cells in a time lapse and then multiplying. So unlike chemotherapy, CAR T cells can recognize targets, kill them, expand, and in some cases persist, though it turns out that that persistence may not be as critical as we thought it was originally. The effects of these CAR T cells in clinical trials have been remarkable. I know that you've heard about them from other people, but this was from our paper in the New England Journal of Medicine that was from the ZUMA-1 study led by Sepa Nilapu. And this was a patient with absolutely refractory lymphoma, and you can just see all of these black spots are lymphoma in basically every nodal group above the waist. And you can see at month 3, month 12, and month 18, these black areas are just normal uptake in the kidneys and the bladder and the heart, and there's no residual lymphoma, and this patient at month 18 remained free of disease. This is a patient that we treated in Miami, or actually our very first patient that we treated with CAR T therapy. This is a 59-year-old woman who had 6 prior lines of therapy and was ready to go to hospice because she had lymphoma encasing her aorta. She came to us in December 2015, got 1 infusion of CAR T cells and was in a durable remission. Unfortunately, eventually relapsed and died, but 2 years later. So since that and other trials, we've had the approval of Kymriah, which is Tisagen Leclucel, derived from studies from the University of Pennsylvania. Yescarta, the product that I just told you about for leukemia, for lymphoma, and then a number of other products for lymphoma, as well as more recently for multiple myeloma. So we really have had now a number of products. Now, the only other thing I will note here that of all of these products, only one of them is approved in a pediatric setting. So people have not been as interested in developing CAR T cells in the pediatric setting, which is unfortunate because they're really, although cancers are rarer, obviously are equally devastating in children. But this just shows you the number of approvals and the number of trials, and I can't go into detail there. The Center for International Blood and Marrow Transplant Research started an immunotherapy data registry in part through funding from the NIH. I sit on that steering committee. So far, over 7,000 recipients have been reported to the registry through 2021, and over 7,500 infusions. About 1/6 of those are non-commercial products, that is research products, and the rest of those are patients who are treated in standard of care approved settings. Importantly, we're already seeing an evolution in the way other therapies are utilized. If you look at CAR T recipients initially in 2017, most CAR T recipients for lymphoma had a prior auto transplant in the green, and some of them had a prior allogeneic transplant. And now you see a small minority of patients, and this is even before the approval of CAR T cells in the second line. I'm going to talk to you about that. If you look at ALL, you can see that fewer patients have had a prior allogeneic transplant. So you can see this moving up as a line of therapy. So a critical thing is, how have patients done in the real world? Well, it turns out that about at least half of patients who are treated with commercial products actually wouldn't have qualified for the pivotal trials. They're older, sicker, have more comorbidities, and have, more, are less likely to have been seen in the academic centers and be a highly selected group of patients. And yet, long-term follow-up, again, using these registry studies, suggests that outcomes are basically the same as they were in the pivotal trials. Unfortunately, that means that this is not perfect. About 40% of patients who receive axi-cel are alive and well at 2 years, and it's also 30 to 40% for the other two products that are approved. So while this is great, because these patients would normally have had less than a 5% chance of survival at this stage of disease, we still have a long way to go. More than half of patients are relapsing and dying. Recently, the ZUMA-7 study was published in the New England Journal of Medicine, as were these other two studies, the Belinda and the Transform studies. These looked at high-risk diffuse large B-cell lymphoma who were refractory to first line of therapy or relapsed within 12 months of the first line of therapy. Here, the relapse within the first line of therapy, normally these patients would have gotten salvage chemotherapy and an autologous transplant. So they were randomized here between CAR T-cell and salvage and auto transplant. Now, this is important because the patients did not all receive salvage therapy. They were randomized before salvage and auto transplant or CAR T-cell therapy. And you could argue that patients on this arm might have done poorly if salvage was poor. And indeed, less than 40% of patients actually got to the autologous transplant because salvage chemotherapy following relapse is not very good. So this isn't exactly an apples to apple comparison saying that auto transplant isn't good, but auto transplant, the way it's done, appears to perform inferiorly to CAR T-cell therapy. So what did these trials show? Well, two of these three randomized controlled trials favored CAR T-cell therapy in the second line. The third did not. RCTs demonstrated that traditional salvage therapies are suboptimal. Less than 40%, again, achieved a PR and went on to the auto transplant in the transplant arm. But retrospective analyses suggest that people who did achieve a PR with salvage do quite well with auto transplant. So additional data including from registries like the ones that I told you about are needed. But for now, patients with early relapse, that is less than in a year, second-line CAR T-cell therapy is the new standard of care. Additional randomized controlled trials between products and optimized auto transplant approaches would be helpful, but are very unlikely to happen. What about third line? Well, we need better therapies following CAR T failure. Long-term results of all three commercial products suggest 30 to 40% cure rates. But CAR T trials after failure, that is second CAR T therapies, demonstrate modest response rates, less than 30% overall response rates. So we need better second-line CAR T therapies. Recently, first-line therapies, the ZUMA-12 study published by, presented early on by Satva Nilapu and all, have demonstrated encouraging results in the first line, but demonstrate responses similar to R-CHOP or traditional chemoimmunotherapy, and these are highly selected patients. So I think we're going to see things move up, but we're not seeing dramatically different outcomes or 100% cure rates in earlier settings. I just say this because these are registry data suggesting that patients who have an allotransplant after lymphoma with relapse, this is acute lymphoid leukemia, can be cured by, people who relapse after CAR T therapy can be cured with an allogeneic transplant. And indeed, patients who went straight to allotransplant have a high cure rate. So whether we should leave people alone or whether we should consolidate with them with an allotransplant is also something that we don't know, but largely we're not doing this as standard of care. So in the last portion of the talk, the next portion of the talk, can we predict failure? Well, there are reasons for failure. We could have short persistence, which could be T-cell intrinsic, or we could have rejection. And we can also lose the target on the cell surface. We can lose CD19, for example, through deletion or mutation. And indeed, this has been shown in pediatric settings, as well as retrospective analyses that were done by Jay Spiegel, who I recruited at the University of Miami from Stanford. So a number of approaches targeting multiple antigens, like CD19 and 20, CD19 and 22, CD22 alone, or 19 and 22, have been done to try to get at this issue of CD19 downregulation in people who have relapsed. It turns out, and Jay Spiegel showed this in a Nature Medicine paper last fall, that immunohistochemistry at baseline is not associated with the likelihood of responding to CAR T therapies. Well, in this trial that they showed, where they had only a 30% response rate to a CD19 and 22 CAR, they did show something interesting, and that's that when they used flow cytometry to characterize the number of CD19 molecules on the cell surface, that most of their relapses came in patients who had very low CD19 on the cell surface to begin with. Of the patients who responded, only one patient failed therapy. Or I'm sorry, patients with higher density, only one patient failed therapy that has had progressive disease, where again, there was a positive correlation between the amount of CD19 on the cell surface. So there are a number of issues I think that we need to think about. And I'm not going to go into detail, but this is from a review that Jay and I wrote from a model proposed by he and David Miklos. We can look at the product fitness, CAR T construct and manufacturing, tumor-associated events, and then the pharmacokinetics, and are the cells persisting? And there are a lot of exciting things happening at UCSF. Julia Carnevale just had a lovely paper in Nature suggesting an approach to increase CAR T cell persistence in murine models. Well, what therapies should we expect? Allogeneic CAR T cells are promising. Other targets are being explored. NK cell therapies, natural killer cell therapies, including with a CAR T cell to engineer them and direct them, are being approached. But we need more development for rare indications in pediatric populations. Increasingly, regionally manufactured therapy are in trials, including multiple active programs at UCSF. So the last portion of the talk, I want to talk about access and equity. So this is a slide I just got from the National Marrow Donor Program this month, talking about hematopoietic transplant. Basically using market demand calculations that were based on the SEER cancer registry data about the number of patients who have diagnoses that should be treated with an allogeneic transplant. They can calculate the expected need in young adults and older adults. I hesitate to say older adults because I'm in this group. And they looked at, for me, the number of transplants that were actually done in the San Francisco area, that is done at the centers in the San Francisco area, Northern California area. And you can look and notice that there was a local unmet need, 152 patients who were under the age of 55 and over 300 patients over the age of 55. And the local unmet need for 2019, the most recent data that we have available, was 454 adults. So we're doing less than half of the transplants that should be done in the area. Well, who's not getting transplant? Unfortunately, traditional barriers like socioeconomic and racial barriers are important. This study that was published in Transplant and Cellular Therapy showed that white patients were more likely to receive an allogeneic transplant than Black and African American patients, 45 and 27% of patients. And patients from areas with higher poverty were less likely to get transplant. Is CAR T reaching everyone? Well, it's obviously revolutionary. But the short answer is no. There are 30,000 diffuse large B-cell lymphoma patients diagnosed annually. About 10,000 are relapsed and refractory. And on the basis of the available market and registry data, it's clear that no more than 30% of this eligible population has received an approved product. Well, why? Well, these are data from Cellular Health that I just received, suggesting that distance to the center matters. In the Southeast, especially the Deep South, where the rates of CAR T therapy are lower, there is an increased distance, for example, 46 miles on average from the patient's home zip code to the center where they received CAR T therapy. This fits well with the UCSF vision to bring care closer to patients, where they need it. Unfortunately, there are other issues. If you look at, for example, a distribution of all patients with CAR T cell therapy, you can look here at the dearth of African American patients. African American patients are about 13% of the US population, but across these registries and clinical studies, they're only about 5% of CAR T patients. And the same appears to be true of Hispanic and Latinx patients. So other disparities in pediatric populations. Black patients had more prior lines of therapy, more relapses before CAR T. In the Tisagen Leclucel pivotal trial, 34% of Black patients in the cohort, 4 of 11, compared with only 6% of white patients of other ethnicities, didn't receive their infusion. They had a lower CR and lower overall survival, probably because they were treated at later stages of diseases, and we need to understand the reasons for this. So what do we know about access and equity? All CAR T therapies in aggregate are underutilized. High-cost tertiary or quaternary therapies tend to maximize these barriers. Early data suggests that African-American patients are less likely to receive CAR T therapy and may have lower overall response rates and CR rates. And unique access issues exist for pediatric patients for whom fewer options exist and who are all primarily covered by Medicaid. And the cost and complexity of access and care compound these historical barriers. So this is a slide I love. This is from Jeff Oletta from the NMVP. Of all the forms of inequity and injustice in healthcare is the most shocking and inhumane. This is Martin Luther King. So how can we improve access and equity? Well, from diagnosis to referral, to collection, to infusion, to no lethal toxicities or no relapse, we have to think about streamlining and scaling manufacturing, better educating patients and referring physicians, developing less costly therapies, and then advocating and meeting patients and referring physicians and addressing these issues. So finally, we can see that we have gone a long period, 125 years or so, with really no effective therapies for leukemia. We started to enter the combination chemotherapy era, the transplant era. We understood that transplant was an immunotherapeutic modality, and we're increasingly exploiting that and improving outcomes. And finally, in the last 5 years, we have the approval of CAR T therapies. So we have gone from no effective therapies to the chemotherapy area, to the combination immunotherapy era, and I think our future is increasingly effective immunotherapies. So I have a lot of people acknowledge. I really want to acknowledge from UCSF, Mike McCune, Mark Schumann, who recruited me as a fellow, Bob Wachter, who persuaded me very passionately to come back, and the faculty and the senior leadership of the division and the center. These are the many individuals. I tried to note them as we went along, and individuals for whom I borrowed slide with and our funding sources. And I'm going to stop there. Thank you.
Krishna, that was just terrific. Lots of comments in our Q&A about how elegantly and eloquently you explained this very complex topic. And as someone who sometimes just really has to make sure I'm understanding all the signaling pathways in the mouse models, I followed that with ease. So really appreciate it. We have a number of questions, and I am going to unify it with a question that really came out of your talk for me that sort of seems to get at a lot of the questions folks had. I was really interested in this idea of moving to CAR T with no prior transplant. And I just keep thinking about the analogy to the changes in how we treat ulcerative colitis and Crohn's now, using early biologic agent like infliximab before using immunomodulators like azathioprine or methotrexate. And I'm just wanting you to look in your crystal ball and think, is that how we're going to use CAR T in the future? So Mark Schumann had a question in the Q&A saying, are there biomarkers predictive for the responsiveness of CAR T? And can we find a population that doesn't even need to get traditional chemotherapy and go straight to CAR T? And what are the barriers to that? Is it cost? Is it access? Or is it something about the therapy that we actually shouldn't use it as first line because of potential toxicities or the potential to ruin the response to downstream treatments? There's a lot in there, but I was trying to unify.
There's a lot in there. So let me address the last part. I've actually been working very actively, and I was actually one of the two outside experts for ICER's analysis of cost and quality for CAR T cells. And I've been part of an MIT think tank for 7 years looking at financing of curative options. The financing part is hard. And I think that, CHOP is actually a very cheap therapy. We can cure patients 70% of the time for probably $30,000 to $50,000. And most of that's the antibody, the rituximab. Second line therapies, auto transplant's not very expensive. It's about $100,000 if you really look at things, and outcomes have less than 1% mortality at all centers. CAR T have 3 to 5% mortality in most trials. It's a little bit lower now. So I think that there are things that we have to think about. We automatically reflexively think of transplant as kind of, draconian and, barbaric, but I'm not sure that it's actually less toxic, even though obviously there are unpleasant side effects and people lose their hair and do have, some limits. So I think we have to address this, and that it's a really difficult question to answer because we aren't going to have data comparing products, and we don't have sequencing data. Sequencing meaning what do we do first? answering Mark's, I know how Mark thinks, and I think he thinks biologically. I think that the data from Stanford looking at, for example, quantibrite, or looking at the number of molecules on the cell surface as opposed to things like immunohistochemistry. In other words, is something CD19 positive or not? But rather than saying, well, how bright is the CD19 on the surface, might predict the patients who are best to respond. Obviously, precision medicine and genomics are telling us subsets of disease that are less likely to respond, though it looks like both auto transplant and CAR T can cure these, double hit and triple hit lymphomas, double expressor lymphomas. I think another way to look at this is cell-free DNA, circulating tumor DNA. The Stanford group and Ash Alizadeh has done some lovely work in this area. Can we predict who hasn't gotten a complete response and then therefore is likely to respond before they respond in a way that isn't easily treated? I think these are all critical aspects. But I think it's going to be really hard, because at the end of the day, how do you do combinatorial trials? So we're going to be, and of course, manufacturers are going to do the trial that they think will win and get them approval, right? So, and that's natural. And so those of us who advise companies, I think, have to, make sure that they're doing the right thing and we're not throwing the baby out with the bathwater and abandoning approaches, because the old approaches are actually very effective. If you look at the Zuma 7 second line data, cure rates don't appear to be better, even though CAR T was better than auto transplant. We're not curing 70% in that setting. We're still kind of at that same place. So I think that the jury is still out. The Zuma 12 results in the first line don't look better than CHOP. I mean, they look great, but they don't look better than CHOP. And I think they're highly selected patients who got to MD Anderson and other places. So I think this is really, we need more data.
Excellent. Thank you so much. That really helps me, at least, and hopefully the audience sort of put CAR T in, sort of helps with that sequencing, as you said, of thinking. And we had some questions from people who are referring folks here for clinical trials and thinking about, when do you really need that clinical trial? When do you go with the standard of care treatment? And it sounds like what I'm hearing is the standard of care treatment is still the first line standard of care treatment, because not only the cost and access issues, but also they are effective and they work in
the first line. R-CHOP is still standard of care. But again, the thing that I would recommend is that patients who, insurance companies are slow to approve, there are delays, and lymphoma, when it progresses, can explode. These are not like myelodysplasia or even leukemia with cytogenetic abnormalities, where you often have months in which to act. So I would actually say, and I know that Babas Andriadis and others who are leading the lymphoma program here want to hear from people. We just recruited two very bright people, Madhav Seshadri and Michael Spinner from Cornell and Stanford. They want to be on the phone with you and are happy to give you advice about whether I need to see this patient tomorrow or whether you can wait a month or two. And I think that's the kind of dialogue that we need to have with our referring physicians, and is critical, because options and trial availability changes sometimes on a weekly basis, and we have new data.
Awesome. I think we have time for one more question. A very good one from Candy, I'm sorry, Sorunets. She's PharmD here. She's sort of pointing out, how do we really engage the CAR T cell pharmatherapeutic manufacturers in improving this access? What are the barriers, especially for underrepresented patients, and how would you imagine we'd go about tackling that?
I think we have to put pressure. So the first thing that I would say with respect to the price issues is we have to focus on value. Costs, things that are expensive, if you're saving lives with many years to be gained, most average chemotherapies for cancer are $10,000 to $15,000 a month, and most of them kick the can down the road a couple of months. These are curative therapies, so even though they've been priced very high, disappointingly high for many of us, they do have value as assessed by the kind of people who says, well, is this worth it, and is it worth it relative to other things? But we do have to engage people as stakeholders working on. I had a call with the director of ambulatory payment models at Medicare a month ago, and the general accounting office just reached out to us. We're going to have a call next week to talk to us about regenerative medicine and what we think about costs from a society perspective. So I chair the government relations committee. Pharmacists, physicians, patients can get active on and put pressure both on manufacturers and Medicare and payers, right? So I think we have to work together. We want to see access, but we don't want to stifle innovation either. So it's tough, and we have an enormous possibility, but I also do think there are 120 companies that are developing CD19 therapies. So that competition is going to drive costs down. Allogeneic CARs, which are coming, I didn't have time to present the data, can be produced at massive scale from a single or two products and don't have to be manufactured in a bespoke way. So the marginal cost of manufacturing should be way less. And they're looking good in preliminary trials, not as good as auto products. So those are all ways I think that we can push the field forward and improve access.
Terrific. Thank you so much for joining us today. I learned a lot. I hope you all did too. Join us next week. Take care, all.